I'd now like to talk about a very important aspect of embryology and developmental biology, what is termed specification. Basically, specification is having cells form and tissues form from an undifferentiated state. They're being specified at a very early stage to start dividing, to start going in a direction of differentiation that was actually put in there by the nucleic acids from the egg. And then as fertilization took place, the zygote forms and it turns on these various factors that will start differentiation to take place, basically mostly under the control of what was put in the egg by the female, by the mother. But let's talk about specification then, and we can talk a little bit about early cell commitment and early embryonic development. One of the first American embryologists by the name of William Keith Brooks in 1883 wrote that, and I quote, "The greatest of all wonders is the existence of a simple, unorganized egg and its power to produce a definitive adult animal." He goes on to say, "We may fairly ask what happens during this discovery of its solution, of reading its true meaning, its hidden laws, and its causes." Well, it's been 130 years since William Keith Brooks made that statement, and a lot has been done to answer some of those hidden questions and hidden laws and hidden causes that he referred to many, many years ago. And I think whether an animal becomes a frog, a fish, a fly, whatever, many of the mechanisms that he's referring to from the egg onward is really an amazing phenomenon. But it's something that we can start to understand, and it's something that hopefully you will understand more as we move forward with this course in developmental biology. Your book talks about levels of commitment that lead to the generation of specialized cell types or differentiation. These levels of commitment or processes of commitment could be divided into three categories. One is specification. We already mentioned specification. One is determination, and the final one is differentiation. So just to redefine slightly, specification is the fate of the cell or tissue that can differentiate autonomously. And if we think about what is programmed in that egg by the mother, very early development takes place through specification. And if you take some of those early cells that have formed, although activated by the sperm, they're under the control of the maternal genome during early development. And if you put some of those early cells in a petri dish or a test tube, they will go ahead and differentiate in that neutral environment. In other words, they have already been specified to do certain kinds of things. And at this stage then, cell commitment could still be changed, could be reversed. And that comes to the next component of differentiation, preceding differentiation, and that's determination. Determination is the second stage then really of commitment. It's where a cell or tissue can be influenced by outside factors, paracrine factors, inducing factors, and it can be determined then to form in a certain way. You can say take certain tissues that would ordinarily, let's say, become flank tissue in the side of a frog, transplant them to another area of that frog, and they might become mouth parts of the new frog. Or you could take neural crest cells that were going to innervate the gut, and they will become pigment cells that would innervate the wing. So, in other words, this is a process of determination. Once, however, they have been determined, once they have been influenced to differentiate in a certain direction, then they are determined. And you could take those cells, once they have been induced, once they have reached a certain level of being induced to form something by these paracrine factors, they then are determined, and you can put them anywhere and they will differentiate in that direction. Take, for example, the eye of an amphibian. If you take the very early eye of an amphibian, the side of the head, basically where the lens forms, and you transplant that skin at an early stage to the flank, it will become flank skin. If, however, you wait until induction or determination has taken place by being induced from the brain stem, that skin will then, if you take it and transplant it to the flank, that will still turn into an eye. we can say that it has been determined to form into eye tissue. And finally, of course, and that leads into the next phase, development, differentiation. This is the formation of the final tissue, and the eye would be an example of that. But it's something that's required by the organism to function, whether it's turning into muscle tissue or nerve tissue or gut, tissues in the endocrine system, and so forth. So the types of specification then we can talk about would be also changed. They would be autonomous, conditional, and syncytial. But before we do that, let me just call your attention to this chart here. These are the various kinds of differentiated cell types that we get, and you can see that they are very broad. Various kinds of blood cells, pancreatic islet cells, which will produce hormones, Leydig cells, cartilage or bone-forming cells, muscle cells, various kinds of organ cells, and all of this started as a simple undifferentiated tissue that was in the zygote, and it's these that had become determined to go in this direction. And although very early development takes place from being committed from specification, in later development then it becomes determined by the induction of using induction factors, using growth factors, using paracrine factors. Now, if we talk about specification, specification has several different types of specification, and we'll go on and we can talk a little bit about this. The types of specification include autonomous specification, conditional specification, and syncytial specification. So let's go through what each of those kinds of specifications refers to. If we take first autonomous specification, certainly classic example is here in the blastomere that has been from the patella, and that's with regard to the types of specification, they include autonomous specification, conditional specification, and syncytial specification. And they're different, and in fact at different stages specification may change. But let's talk about autonomous specification first. Here is an example using the snail, which is genus Patella. Basically, when the snail embryo starts to divide, it first has cells that will undergo division, and certain cells, called the presumptive trochoblasts, are destined to form cilia. And you can take those cells at a very early stage, you isolate those trochoblasts, and they will go ahead and they will form the specialized cells that contain the cilia, as you can see down here. Just the same as if they were left in the intact embryo, they will form those same cells up here. So that says that there's autonomous specification. In other words, those cells will autonomously form cilia and become that specialized kind of trochoblast cell no matter where they're placed, whether it's out culture dish or whether it's left intact in the developing embryo. Autonomous specification. Another example of autonomous specification is seen in the tunicate embryo in terms of the formation of the tail muscles. So here you can see in this example, this is now in the tunicate embryo, the C-squirt. Here at this 8-cell stage, there are cells that do certain things already because they are specified to make those kinds of tissues. Here's the animal pole, the vegetal pole. Ectodermal cells will form from these cells. Nervous system will form over here. Notochord, and then endodermal tissues will form here. And also a very special group right over here, this part of the cell, of this mesenchyme, will also then form muscle tissue. And so if you isolate these cells at this 8-cell stage, they already contain the messenger RNA placed in those locations by the mother in the egg, and they will basically then form into what they were destined to form into. Here B4.2 forms into ectoderm, A and B. 4.1, notochord tissue along with endoderm. Here's the muscle tissue that forms from the B4.1 along with endoderm. So if you isolate these cells, taking as an example B4.1, you will end up with muscle tissue and endoderm, And that's because those are placed in there even before the first cell divisions have taken place in this C-square. Now, if you take these cells at the 4-cell stage, actually, and you take a pin or a needle, and you just press the site of the cytoplasm here, and you force some of this yellow cytoplasm that's going to be forming the muscle tissue from the B4.2 blastomeres up into the animal pole, then you let these go ahead and divide and differentiate, and you separate them. The tissue included in the ectoderm will include muscle tissue, and the tissue that's included in the original endoderm with the muscle tissue also will include muscle tissue. So, what that illustrates then is autonomous specification. Now let's talk about conditional specification. Conditional specification is observed in this amphibian embryo, and what that suggests is that's the ability of cells to achieve fate by interacting with other cells. In other words, those cells, if you took them out, they're specified. If you took those cells out, put them in culture at an early stage, they would go ahead and form back cells that they form into in the embryo. If, however, you transplant those cells to another neighboring area that will receive inductive factors from that new area, those cells will form into cells that correspond to the new area in which they are transplanted to. So if we look at this illustration here now, we can see that here's the blastula stage, the animal pole is here, vegetal pole here, and if we take some normal back cells, transplant them into the belly area of what is going to be the pre-belly area, you can see that what happens is the cells will incorporate initially into the back part of the embryo and then into the belly part of the embryo. If you take and remove certain portion of the cells, as long as it's not too big of a portion, and with a glass needle, you can then have that embryo, that blastula stage, will reform that area, and you will get a totally normally developing embryo. In the late 1800s, there was an individual by the name of Weismann from Germany who proposed the germplasm theory. And what Weismann's germplasm theory of inheritance said was basically that any other cell other than a germ cell did not contain all of the determinants, did not contain all of the genetic information. Well, we know that's false. That's not correct, because we know from the work of on Dolly the sheep, the experiment there, that you could take an adult cell and form an entire new animal from that adult cell. However, what you see here then is an example of what was proposed at that time. That was in the late 1800s, and Professor Weismann basically said that unless it was a germ cell which contained all of the genetic material, from these germ cells you would get cells dividing that would contain only part of the genome. So some of those would contain the chromosomes for muscle, some would contain chromosomes for blood cells or for nerve cells, and they would then basically these things could only come from germ cells originally, but once they had branched off of the germ cells, then they could differentiate into somatic cells because they had the genetics to be able to do that. Of course, as I said, this theory is incorrect, and there were some people, contemporaries actually at that time, that did some experiments that showed that this in fact was incorrect. One of those individuals was Wilhelm Roux, actually, who was working on it. Although he didn't disprove the theory, he actually gave evidence that supported the theory, although later on this evidence was found to be incorrect. But basically what Wilhelm Roux attempted to do to demonstrate autonomous specification, in fact, didn't really do that. What he did is take a fertilized egg that went to the second cell stage, took a hot needle and killed basically half of the embryo at that point, one of the two cells. That dead tissue then was left intact against the living tissue that continued to divide, and he ended up with a half an embryo. And so his conclusion at that, it was a short-lived conclusion because of some other experiments on other species, but basically what he had found or had concluded, and it was a reasonable conclusion based on the data, was that only half an embryo formed. So what that said was that at this stage, each of those contained the information to do half an embryo at the 2-cell stage. Then another German scientist, Hans Driesch, did some experiments which, using the sea urchin, which would disagree with that. Basically what Driesch found, this was in the late 1800s also, 1892, he dissociated sea urchin embryo at a 4-cell stage or an 8-cell stage. This illustrates the 4-cell stage experiment, basically, and separated these into 4 cells, and he found that each one of those cells would develop a totally new pluteus, which would then go on to develop into an entire organism, an adult organism, normal. I mean, each of the plutei were a little different. They were showed variability, but in fact, they did develop into an adult organism eventually, and this then disagreed with the Weismann germplasm theory of inheritance and really disproved it, at least certainly in the sea urchin eggs and sea urchin early development. Another experiment which Driesch did was to basically confirm the idea that a nucleus could be shifted around and could still form a normal embryo. And what he did then was to take, again, sea urchin embryos at an early stage, at the 4-cell stage, and he put those sea urchin embryos under glass slides, put them between glass slides, so that when they divided, they had to divide from the north-south perspective rather than equatorially. And when he let them divide, what he found was that if the cells divided normally, normal cleavage, you would get, for example, cells 5 and 6 here, 7 and 8 here, and if you then put them under pressure, they would end up in different places. Here's 5B and 5A instead of 5A and 5B being here, like would be normal. And so it mixed up where the nuclei were and where the cells were. Then, in this grown under pressure, he then released the pressure to allow the equatorial division to take place. And what he found was that these sea urchins would develop completely normally, and there was no problem with their development. Again, confirming conditional development in sea urchins, and further disproved the germplasm theory of Weismann. This was also then finally disproven in vertebrates, in the amphibian in this case, by another, you guessed it, a person from Germany, Hans Spemann, in 1932, some almost 50 years later. What he found was that if he took an experiment in which he would ligate the amphibian developing egg and let the nuclei be only on one side, and if you let that go to the 16-cell stage, and then loosen the loop so that a single nucleus could come into the other cell that didn't have any nuclei in it, and he let these undergo development, what it did was to end up forming 2 totally normal amphibian embryos. In this case, they're triturus embryos, they're salamander embryos, and that proved then that that single cell from the 16-cell stage was able to form an entirely new embryo, an entirely new animal, and therefore that nucleus contained all of the genetic material required for normal development to take place. And so, again, showing that the Weismann germplasm theory really was incorrect, even though it was a great idea at the time, a great question that stimulated a number of exciting experiments. Definitely it proved to be an incorrect theory. Let's go on then to conditional specification, and that's when we talk about conditional specification, we're talking about how morphogens can react in different ways or can treat cells in different ways and make different cells from the same initial morphogen depending upon the concentration of that morphogen. So if you take a morphogen and you basically put it in and see how it will diffuse, you can find that the amount of morphogen, of course, closer to where it's being produced is much higher. There's a threshold level that it may have that causes a certain kind of cell to form. If it's a farther distance, there may be a second threshold to make a different type of cell form, and maybe in the third case, if it's below the threshold level, nothing at all will happen. So if we look at that, what we can see hypothetically then, taking activin as an example, and covering beads with no activin or activin to show this kind of differentiation expression. And what they found is when the beads were there with no activin, the cells remained unspecified. If the beads contained 1 nanomolar activin, a low concentration, then a certain number of cells would form that expressed the XBRA gene, in this case. And if you then took beads that had a much higher concentration of activin at a distance from those beads, you would get the XBRA genes. However, as you move closer then to the beads, you would start to get the goosegoid gene expression. And so basically, depending on how far and what concentration you had of this morphogen would depend on what type of cell you would get, whether it would be expressing goosegoid, XBRA, or no expression at all. The final type of specification I wanted to talk about is syncytial specification. Syncytial specification is a combination of autonomous and conditional specification. Insects, for example, use this certainly in the early stages of development. The insect nuclei will divide but not the cells, and so there are many nuclei within that insect egg that is being influenced by materials, morphogens that had been put in that egg by the female, by the mother. And so those nuclei, depending upon where they're distributed in that syncytial component of the developing embryo, would depend on what they will end up expressing. So in Drosophila, there is a gradient between the anterior and the posterior portion of the embryo, and those genes that are toward the anterior end will form head and thorax type structures, whereas those toward the posterior end will form tail and abdominal structures. And these will then be expressed as the embryo continues to develop. They will be determined to form in a certain way as cells initially around the outside of the developing embryo, and they will then form into the specific component parts of the body based upon what their morphogen exposure was in this syncytial type of specification. So, just in summary, each of the 3 strategies of specification, autonomous, conditional, and syncytial, basically have a different approach, although they often work together as the animal goes from an early stage of development into the adult stages. And each of these have a different way to provide the embryonic cells with transcription factors which will activate certain genes and cause those cells to differentiate in a certain direction and into a particular cell type. Autonomous specification, there the transcription factors are already in the cell cytoplasm, such as in the egg. Conditional, that's determined by paracrine juxtacrine factors, which form neighboring cells. And the syncytial is by interactions between regions of the egg cytoplasm and the factors that those regions within that cytoplasm contain. And together, these types of specification will allow for the various modes of cell types to form.